talk about the chapter 10, the sensory physiology, and we have some topics to cover here. That's general properties, somatic sense, chemoreception, smell and taste, the ear about hearing, in the ear about equilibrium, and the structure of the eye. So let's first go to the general properties. This chapter in this beginning talks about energy transformation, sensory transduction, neuroreceptive field, central nervous system, sensory information processing, and stimulative properties. So the first one here is receptors are sensitive to particular forms of energy. So each receptor in the body, each neuron has a function that they need to recognize some kind of stimuli. So they will recognize depends on the outside stimuli they receive. So then neurotransmitters will be released by the neurons onto sensory neurons, initiating an actual potential that will send the information to the brain. Okay. Sensory transductions convert stimuli into graded potential. Receptors are specific to receive one kind of energy, one type of energy, but they can receive another types too. For example, a sensor that will receive pressure or touch, they can receive the information about heat, but it depends on the intensity and the duration of the stimuli. Sensory neuron has a receptive field. So what it means, each neuron has a field, has a field to receive the information. So they combining together, they form a bigger area to receive the stimuli. So they combined will get will cover a bigger area, and then will send complete information to the brain. So it will increase the accuracy of the reception. The central nervous system integrates sensory information. So every stimulus that the body will receive will be sent to the brain, and to be computing and analyzing how they need to proceed, how the body will proceed about this stimuli. Coding and processing distinguish stimulus properties. In this case, we have here a schematic figure about hearing, and the sounds just not to decode information about the waves, but also to recognize position and has other functions. And how they will recognize the kind of stimuli. We have the nature or modality, or how can I say, the type of the stimuli, and its location and intensity and duration. All this combined together will send the correct information to the brain. Okay, I will have a schematic figure too that was primary sensor and secondary sensors. So in this case, there is a pin making a pressure in the skin. So if you look at the graph here, we saw that the secondary neurons are activating here. They are activate, and the primary neuron is has more intensity. So when pass time, they will turn off the secondary neurons, okay, and will feel just the primary neurons. So the other ones are deactivated here. So they will feel, because of the time, they will feel more intensity in the stimulus. So another schematic figure about the transduction. Transduction site, trigger zone of the neuron, myelinated axon, cell body, and axon terminate. So here's another graph that talks about the duration of the stimulus. So here the durations, there's a lower pressure, and there's not that big time. So, and the other one has a longer time of the stimulus, like If you pressure your skin, you just touch, or you just pressure, or just keep your finger in the skin, the stimulus will be recognized in different ways to send the correct information to the brain. Okay, so now we go to the somatic sense. Pathways for somatic perception project the cortex and cerebellum. So there's the somatic cortex, is the part of brain recognized where extending sensory tracts originate. Each sensory has a corresponding region in the cortex that will censor pathways to the left hand, for example. How can the brain can recognize where is the stimuli? They will recognize, for example, a left hand will be computed on the side of left hand, and from left foot will be recognized by an area in the brain that cares about this location. Some experiments done with non-humans animals shows that the more sensitive is the region of the body, bigger is the area that covers this part of the body in the brain, and it's not fixed. Because if some part of your body is more stimulated than other ones, they will grow in the area that controls this part will grow in your brain. So this is not fixed. So it's very adaptable. Touch receptors respond to many different stimuli. So as I said in the beginning, as I said, if you touch your skin, the sensors will feel this touch, but they can feel pressure, they can feel heat, they can feel other kind of stimulus to send the information to the brain. But it depends on the intensity and duration, as I said in the last slide. Okay. Temperature receptors are free nerve endings, and it's interesting because when your body tries to regulate your temperature, and they will check about 37 degrees, that's your normal temperature of the body, to 45. So this is just to control your heat, your temperature. But above 45 degrees, they will activate pain sensors or pain neurons receptors that will send to the brain information that some kind of injury can damage your skin or tissue. So it will pass from just to sensor and will release some kind of pain. So you need to make some response to don't get injury in the tissue. So then nociceptors initiate protective responses. So about this information that he retrieved about these neurons, they will send some information to the brain, and then the brain will respond to what we're going to do. What do you need to do? Some reflect to, for example, when you touch a hot surface, you need to just your reflex will move your arm far away to the heat surface. So there is a kind of response that the brain will give to you. And the nociceptors respond for strong notions stimuli. So as I said, strong notions, chemical, mechanical, or thermal, as I said, it's a response from the brain about this information that he gets from the sensors or from the neurons. Pain and itching are mediated by nociceptors. What it means. For example, indicating three interesting points about the pain is that it's highly individual. It can change to one person to another person, and so it can vary about your emotional state too. And other interesting point is that he can be magnified with your past experience, or he can be suppressed. For example, when pain cannot stop you. For example, in case of survival, it depends on the ignoring injury. So if you are in a battle, you get hurt, but you cannot stop because you need to keep alive. So your system will ignore injury and keep going. And pain in internal organs or visceral pain, partly localized. It's difficult to say where the pain is coming from. So there is some place that the pain will reflect. So, for example, in cardiac ischemia, maybe felt in the neck or down left shoulder or arm. So it's not exactly in the place, but it's someplace around. Okay. So here is just for information, a gate control theory that will adjust your neurons, and they will sometimes turn off this connection to partial inhibition or ascending pain pathway. For example, when you just hurt yourself, your skin, and you start to rubbing, it will activate another kind of stimuli. It's not the pain, but you activate the touch stimuli. So two stimulus at the same time can kind of confuse the brain, or they will, how can I say, they will have the responsibility of the neuron, so they will turn off some kinds of pathways to make you feel less pain. That's why when you rub your skin, when you hit, you can feel less pain. This is a schematic view like this. Noxious stimulus, and here the noxious stimulus plus the touch or non-painful stimulus, like the rubbing your surface. It will be two stimulus, and that will activate the A beta fiber and C fiber. C fiber is responsible for the pain, and A beta fiber from the non-painful stimulus. Okay, and also about the pain, we have analgesic drugs that range from aspirin to potent opioids such as morphine. And this will decrease inflammation and presumably slow the transmission of pain. It's important because the transmission of pain would be broken here as another stimulus. So they will simulate the stimuli to break the pathway of the pain. Okay, so we have chemoreception, smell, and taste. Olfaction is one of the oldest senses, and it's not it's a chemical reaction that the olfactor will provide to your brain. So they will feel the molecules in your nose, like here. And so it's more older when animals who survive the pains olfactory cues, and this will extension of the forebrain that receives input from the primary olfactory neurons. But what's occurring at the cellular and molecular levels that allows us to discriminate between 1,000 of different odors? So there's a lot of neurons here that will combine all the stimulus, and like letters form became the word. The combination of these stimulus will be translated to the brain as a knowing kind of odor. Okay, taste is a combination of 5 base sensations too. So it has bitter, salty, sweety. So and this will all combine in your tongue. They will the neurons will sense this kind of stimulus and send the message to the brain too. And we think that only the tongue is responsible for our taste, but actually the nose is a big part of this. So the aroma specifically is the most responsible for your taste. So when you get cold or some problem in your nose, you cannot feel the real taste of food or something like this, because your reception in the nose is not working. So the taste will not be complete. Only the neurons on the tongue and the mouth will not be enough send the whole information to the brain. Taste transductions use receptors, like bitter taste uses about 30 variants of T2R receptors, and some pathways release CA2, CA2 plus from intercellular stores and cation channels and allow the CA2 plus to enter the cell. This is an schematic view about the parts that will sense these flavors and salt, bitter, and each part is responsible for one, like sweet here, and umami, bitter, or sour. So each one is responsible for each kind of this stimulus. So there's another schematic view. When ligands activate the taste cell, and various intercellular pathways are activated. And CA2 plus signal and the cytoplasm triggers the exocytosis or ATP formation, and neurotransmitters or ATP is released. So the primary sensor neurons fires an action potential and sent to the brain to recognize all these stimulus. Okay. So the ear, concerning about the hearing. Hearing is our perception of sound. So our membrane in tympanum will receive the waves that will enter in your ear canals and will be treated like mechanical waves, and will transduce these mechanical waves to a fluid, to a bone, and we'll see here the process that will, until we get the information to the brain in electrical signals. So it's a schematic view here from the ear. So the ear canal, the waves will come here and will be transduced by the tympanum as another vibration here in these parts of your ear, the malleus, incus, and stapes, and this will go to the semicircular canals that has fluid that will vibrate in the cochlea, and the cochlea has a nerve inside that will transduce all these vibrations into electrical signals and sent to the brain by these nerves. Sound transduction is a multi-step process. As I say, there's a lot of process in the ear. The tympanum, the cochlea, and the other parts of the ear will help to transduce the sound waves to electrical signals to the brain. So here is a representation of the cochlea, and here there is some fluid, and this will vibrate, and these nerves will receive this information of vibrating and transform to electrical signals that will send to the brain. Okay. The cochlea is filled with a fluid, as I say, and this fluid will vibrate the nerve and will send the electrical signal. And all this process beginning in the ear canals, passing to the tympanum, and go to the other parts of your ear. Okay, here's another schematic view of the same part of the cochlea inside, and here the canals, the semicircular canals that will has fluid here that will vibrate with the sound waves. Sounds are processed first in the cochlea. So the auditory system processes sound waves, so they can discriminate location and pitch and loudness. And the localization of the sound is a complex process that requires sensory input from both ears. So measuring the time delay about the font of the sound. By this time delay that arrive at your ears, or left ear or right ear, they will give you the location of the sound. And auditory pathways project to the auditory cortex. This is an interesting part because this has a more flexible part and is a more stiffness part. So this will recognize the low frequency and the high frequency. So they vibrate differently and recognize this kind of waves here to send the message to the brain. Here is another schematic view, and the cochlea from left side and right side that will be sent to the other side of your brain. So it's inverted, the signals, like in the eyes. Hearing loss may result from mechanical or neural damage. So in conductive hearing loss, the sound cannot be transmitted through either external ear or middle ear or any way. And there's a central hearing loss that results either from damage or neural pathways between the ear and cerebral cortex of damage, and or from damage of the cortex. And the sensorial hearing loss arises from damage of the structures of the inner ear, and this will cause that the waves cannot pass to the cochlea and be transduced in electrical signals to send to the brain. So there's two types here of problems that you can have with hearing loss. Okay, so let's go to equilibrium. The vestibular apparatus provides information about movement and position. So as I say, all these parts of your ear will call labyrinth. They will recognize your position. So when you, for example, you turn your head to the left or turn your head to the right, or turn down or backwards, there is some hair cells that will feel these movements, and these movements will be transduced in electrical signals and send the message to your brain about your position of your head. So that's why the system will help you to get the equilibrium, because they will recognize the position of your body. The semicircular canal sense rotational acceleration. So as I say, these hair cells in your ear, they will, for example, when you are, as I say, going backwards or going down, they will feel this movement, and they will go because of the gravity, they will move and transform in the electrical signals. The otolith organ sense linear acceleration and head position. As I say in the last slide, is the same thing. So because of gravity acceleration, cause the otolith to slide forward or back. Is a gelatinous otolith membrane slides with them. So when you move, this membrane will move too. So that's why the brain can recognize the position of your head and make it you get the equilibrium. Equilibrium pathways project primarily to the cerebellum. So these vestibular hair cells will release neurotransmitter onto primary neurons, and the vestibular nerve, a branch cranial nerve, will give this information to the brain. Those sensory neurons either synapse the vestibular nuclei of the medulla that go without synapsing the cerebellum, so they go straight to the brain, and which is the primary site of equilibrium process. So this is a representation of the vestibular branch of the vestibulocochlear, and transmitted to the cerebellum and transmitted to the vestibular nuclei of the medulla, reticular formation, thalamus, and cerebral cortex. Okay, so and the last one is the eye and vision. The skull protects the eye. So in the skull we have orbit. The orbit that is a cavity in your skull that will protect your eye, in your eyeball. And axillary structures are associated with the eye include 6 extrinsic eye muscles that will move your eyes, and they are responsible for these movements, and the cranial nerves that innervate these muscles to get the responses of stimuli and etc. Okay. Light enters the eye through the pupil. So the light will pass to your eyes, entering the pupil, and will be passed from the pass for the lenses that will reflect on the background of your eye, and these lenses are not fixed. The interesting thing is that the pupil is not fixed, and pupil size is not fixed, and the lens is not fixed too. So when there is too much light, the pupils will close, and where there is when there is no light, the pupils will open to let enter more light, and this lens will move by the ciliary muscles because they need to focus the light. So they will reshape themselves just to focus the light. So that's what I say, the focus of the light. So why the eye need to focus the light? Because of permeability of the light, they will pass through air, from air to another environment. So when you put something in the water, they will suffer of refraction. So this will distort the light. So you need to adapt from this refraction, and the lenses will focus and correct these problems of the light passing through the other kind of environment. So in a schematic view, when the right eye will be processed in the left side, and the left eye will be processed in the right side. And phototransduction occurs at retina. So the retina will convert the light energy into electrical signals to the brain. Then will send to the brain and will form the images in the back side of your brain. And the wavelength that we can see is from 400 to 750 nanometers. This is the wavelength that a human eye can see. About X-rays and gamma ray, we cannot see this kind of energy. There is another schematic view of the refraction and how the light will behavior when the pass to the pupil and pass the lens. So that's what's going to happen. The light will be passed on the lens and suffer from refraction, and so it will be inverted. So the image will be inverted to your appear inverted to your brain. Okay. So as I say, the ciliary muscles here, you can realize that the lenses are not fixed size, so they will be stimulated to correct this refraction. Here another schematic view when the lenses are moving and the ciliary muscles contracted to do this deformation in the lens and correct the refraction. And photoreceptors transduce light into electrical signals. So we have in our eyes rods and cones, photoreceptors. So the rods will be used for low light and are used in night vision when you have low quality of light or you don't have light. And they are specific for this situation. And cones is high acuity vision and color vision during the daytime. So about the color, and when you have a good quality of light, the receptor that will be used is the cone. And signal processing begins in the retina. So when the light enters your eyes, when the light reaches the retina, it's where they will be transduced from light to electrical signals and be sent to the brain. Okay, my chapter is this, so thank you so much.